I'm excited today to present our research that we started about a year ago with the partnership of CSF and also with Adam Korn, who I learned yesterday is the appointed chair of the Board of Trustees of CSF. And my talk will present our research concept and then will lead to Rebecca Burwell's talk about our first findings in this regard. So, after Dr. Grant's talk, needless to say that certainly we should not be overlooking the cognitive burden that we face in hydrocephalus and associated CSF disorders, which Chiari and Syringomyelia are partnering with. And cognitive burden is still not just a diagnostic challenge, as we discussed earlier, but also in terms of prognostication, we still don't know how to counsel patients in this regard and what to make out of it, bluntly speaking. So in the broader picture, our translational research objects are targeted to develop kind of real-time diagnostic approaches to study and also monitor cognition and cognitive deficits in CSF disorders, but also to find novel treatment aspects. Needless to say, the shunt is not the final real deal to treat cognition in hydrocephalus, and certainly eventually we need new therapeutic measures here. So that is a broader scheme of our research, and certainly that kind of stems or goes back to translational studies that I was doing in hydrocephalus, because when I started as a young resident to be interested in hydrocephalus, it really was, when you went back to the literature, they were always talking about subcortical dementia. Well, what does it mean, subcortical dementia? And that definitely, I felt, had never explained all the cognitive challenges and the different various facets that these patients display when it comes to their cognitive complaints. It's not just the short-term memory. There are much more aspects. And amongst neuropsychologists, I always found friends when I was getting to that. So I was always thinking, well, where does the brain fail? Where is the soft spot of the brain when it comes to failure of cognition in CSF circulatory disorders in a broader spectrum? And I was looking at blood flow studies, not that I was thinking that the blood flow is affected, but I took the blood flow as a biomarker for where might there be a problem in patients. And I correlated the blood flow PET studies with the severity of the cognitive dysfunction using a score, basically the Mini-Mental score, which is not the greatest, but there wasn't anything else at the time. And the more the patients were affected, the more blood flow impairment we found in frontomesial areas, but also in mesiotemporal areas, which you could, for better or worse, attribute to the limbic cortex, which certainly I felt explains a lot what might be going on. And then I was asking, gee, can I provide more mechanistic evidence for what is it? Is it epiphenomenon? Is it ischemia? What might it be or is it some sort of different vulnerability that's going on that affects certain structures in that hydrocephalic brain? And so we were lucky we had a rat model of hydrocephalus, which you can see here on the lower end, models the chronic CSF failure that we see in the patients with ventricles that stay enlarged in the chronic period, some periventricular edema, and normalization of ICP after 4 to 6 weeks. It's a kaolin-induced model of hydrocephalus in the rodent, and we did blood flow studies there using autoradiography, and found that actually what happens in the chronic disease, that the brain kind of strokes out. We found maintained blood flow reductions about between 25 and 50% from the baseline, which is actually a blood flow reduction that is also found in 4-vessel occlusion models. And so we have a silent stroke that ultimately also leads to pathological changes in the brain, and we were looking at not just the hippocampus, but in the hippocampus we found exciting findings of selective vulnerability, which kind of supports the mechanism of a silent stroke in the hydrocephalic brain. And we found in the C1 maintained upregulation of nitrous oxide, which is a marker of ischemia, and hypoxia. So the translational aspect kind of worked, and the more, of course, I'm excited to have that opportunity to go back to looking at doing translational work together with Rebecca Burwell and my other collaborators that I'm presenting at the end. The experiments that we are doing goes again back to the rat kaolin-induced model. And first of all, we want to prove where is the hotspot of the cognitive deficit in hydrocephalus. And we study, obviously, hippocampal circuits involved in cognition and cognitive deficits, and want to prove evidence that these circuits are, in fact, involved as the hydrocephalus progresses. And then also, as we go along, we try to certainly try to identify novel approaches to modify these cognitive deficits by modifying and altering CSF production and absorption at the choroid plexus level and also at the blood-brain barrier level. For the better or worse, actually the kaolin model, and these are some studies that we published decades ago, where we not only looked at the brain but also at the spine. And as you can see here in that radiographic study, is that the animals in fact develop, the arrow points to it, like Syringomyelia. Not a surprise, because with a kaolin model you actually block the cisterna magna, because that's where you inject the kaolin. So it's also a model of Syringomyelia, and some of researchers might disargue with that, but I would say the kaolin model, in fact, might be a good model to study all these circumstances that we see around problems at the craniocervical junction, spine and brain. And we also found that, and that was a discussion Lisa and I had yesterday about that in the spine there is a connection with this lymphatic system as the syrinx progresses. So that there's some certain aspects that we might want to discuss. Anyways, we separated the study or project in two, where one is presented by Rebecca Burwell in a minute, where we are looking, we are recording perirhinal and prerenal areas that are associated with limbic or hippocampal function and dysfunction in the hydrocephalic rats. And then we correlate them with behavioral testing to, in fact, see, is this behavior that declines in the course of the hydrocephalus explained by impairment of neuronal circuits in the hippocampus and in perirhinal areas. And we're doing that by electrical recording, and then to prove further mechanistic evidence to the fact that this is actually relevant for hydrocephalus, we would do those things before and after CSF diversion. Back in Hanover, we developed a technique where we can actually, with a catheter, puncture the ventricles, drain it, and sort of before and after tap test in clinical realms, changes in the electrical recording as well as in the behavior of the rats. So again, Rebecca Burwell presented. Another project that we hope to present in the future is more aiming towards what happens if we modulate CSF production and absorption, and does that help those things to get better, which ultimately gets us to other therapeutic options for CSF disorders other than the shunt or actually surgical decompression. And here optogenetics, certainly a very sexy thing, comes into the play, but very advanced and very exciting, of course. And we will go and try to look at recording and modulation of those hippocampal circuits using optogenetic methods. And as you all know, optogenetic Optogenetics is a method to make neurons sensitive to light. You do viral transfection, and then you can increase channel rhodopsines that can allow to inhibit and elicit neuronal function by green or red light or other light. And we would then see whether we can also optogenetically modify these circuits and whether this actually leads to improved behavior in these animals, which would then ultimately bring up new therapeutic approaches, and also the modeling of the CSF production absorption could then get further evidence to how cognition can be altered for the good or the better in the disease. So basically, in the bigger scheme, we might want to identify biomarkers for the assessment of real-time cognitive function by electrical recording and stimulation and optogenetic recording and stimulation to get the hot spots that might be targets for novel clinical treatment approaches, i.e. DBS or pharmacological or just light. And without further ado, I'd like to present our research group. Chris Moore, Associate Professor of Neuroscience, is expert in optogenetics, and he is collaborating with Maria Latinen from Boston Children's on in vivo and in vitro modulation of choroid plexus function through optogenetic measures, and Rebecca Burwell will talk now. Her expertise is psychological sciences and cognitive and linguistic sciences, and myself. This is in partnership with CSF, as I said, and under the auspices of the Department of Neuroscience and the Brown Institute of Brain Sciences. And I really want to thank John Davenport for being here today and certainly for supporting our research and guiding us. So without further ado, I'd like Rebecca to talk. Thank you.
Well, it's a great pleasure to be here. It's been a terrific opportunity to start collaborating with Petra, and Petra asked me to tell you a little bit about myself and the research that we do in my lab. So I'm going to start with just a little bit of explanation. So we use rodent models, but we use rodent models to we hope to understand more about human memory and attention, and to get a little more face validity. A few years back, we started using an apparatus we call the floor maze, where we back project to the floor and of the maze, so we can manipulate stimuli automatically while rats are doing pretty complicated tasks. And rats are all about the floor. I mean, they really learn very quickly about stimuli that are presented on the floor. And so this is so we'll have this is the apparatus here. This is a floor maze, a projector. We can put any shape arena on top. Right next door is a control room where we can collect electrophysiological data, and we use optogenetics, and we are also controlling the behavioral task. So this is an example of a task that rats learn pretty readily. So you have two objects here. When the object is presented on a gray floor, then one of them will be correct. On a different colored floor, the other object will be correct. So we can look at questions about memory, discrimination, and also executive control and decision making. So we use sometimes food rewards. Sometimes we just go directly to the parts of the brain, the reward centers, and give a little zap for a reward, and that way we don't have to take any time out of the task to go get chocolate milk. We do high density tetrode recording so that we also have integrated optical fibers, so we can combine optogenetics with electrophysiology and behavioral testing. And that's just an example of a drivable fiber, so we can get the fiber where we need it. So Petra mentioned the hippocampus and hippocampal circuits, and this is how we got together. It was our technical skills, but these are the parts of the brain that are affected and probably are the ones that cause memory deficits, and hydrocephalus are the parts of the brain that we study. So perirhinal cortex, which is involved in object recognition memory. These are just a few recent findings. Postfrontal cortex, which is like parahippocampal cortex in the human and primate, in the non-human primate brain, is we see signals that link objects to particular places. Also, all of these regions, perirhinal, postrhinal, and the hippocampus, they all have a strong oscillation, a theta oscillation, and we've also shown that these regions show coherence in the theta frequency band, and especially when animals are looking at new objects. And also the hippocampus is, objects and landmarks are important there, as well as just other episodic memory or memory for everyday events of our lives. So now to get on to what we're doing. So Petra and others have used this KLN model of hydrocephalus. And of course you get enlarged ventricles and changes in gait. But what we wanted to also look at is whether we could find a task where we could see a progression of memory deficits and possibly use that as a model for understanding the cognitive deficits associated with hydrocephalus. And at the same time, we would like to have an electrophysiological signature. So if we could see something that correlates with behavior, something that we can record in the brain, that may give us some idea about how we might ameliorate those changes. So what we're doing is Petra came and taught my research technician how to do this KLN procedure, and she's actually great. She has great hands. She's a great surgeon herself, and she's doing very well. And so I have some data to show you. First, you can't see, these are hydrocephalus patients, and I think you can see here in the rat that you get the same increase in the ventricle size. And the parts of the brain that we've been studying anyway are, so this is the hippocampus in the rat brain, and this is the hippocampus in a sort of a sagittal view of the human brain. And what's happened in the rat is, or in the human, is that the hippocampus it's kind of gone backwards like this, and so that the rostral hippocampus is like ventral in the rat, and the caudal is like dorsal. So we have very good homology of these regions that we're interested in. And they change with hydrocephalus. The volume of parahippocampal cortex goes down, hippocampal volume goes down. After the shunt procedure, hippocampal blood flow increases, and so we think we have a good thing going here. Now, so what would you want in a task if we wanted to have a behavioral memory task in this model? We would want one that we can test repeatedly, and we would also want one where there's not a lot of training. And actually, rats will do something, and actually people, babies, monkeys, we all do this. We like novel things. So this is a task. It's called spontaneous object recognition. You show the rat two objects. They'll explore it pretty vigorously. Then after a delay, you show one of the same objects and then a new object. And a normal rat will explore the new object more than the old object. That is if they remember it. And this is a recognition memory task. These are some of our actual objects. And then this is the arena with a rat doing the task. So what we have done is we have two groups of rats now that are at different stages of normal pressure hydrocephalus. And so I should tell you one more thing. The measure we use here is discrimination ratio. So it's the novel exploration time minus the familiar exploration time. I seem to have a timer going on here, so I'll just get back to where I need to be, if you'll pardon me. Okay, so novel minus familiar divided by novel plus familiar, and that gives you a measure that goes between 1 and -1. So if the rat is only exploring the novel object, it would be 1, but usually it's about 30% for a normal rat. So you would see a positive discrimination ratio. And here's what we saw after we had a set of rats at 2 weeks and a set of rats at 4 weeks. The controls are discriminating very well. There is a significant decrease at 2 weeks, but these animals are still discriminating. At 4 weeks, they're not different from 0. So they're about equally exploring the novel and the familiar object. And then 2 weeks later, when we have them at 4 weeks and 6 weeks, so now these animals are here and these animals are here, where we have really no discrimination. And we've now looked at 8 weeks and still have no discrimination. So the plan is we're working on a procedure for imaging so that we can image during the progression of the hydrocephalus in animals. And we also will be recording. And I'll talk to you a little bit about what we expect to see there. Oops, sorry. Okay. So this is a recording from a colleague. This is Theta in the human brain in an area that's between the hippocampus and the perihippocampal region. So this is what you can see is the raw local field potential, and then the filtered field potential. And right here and here is where you get about of theta. You can see this down here in this spectrogram. Here's theta, and then here's theta, and this is a human brain. Now, hydrocephalus patients have altered theta. And so we are thinking that theta might be, alterations in theta might be our neurological or electrophysiological signature in this rodent model. Now, this is theta in the perihippocampal region and the perirhinal region. This is a coherence gram showing coherence at theta across hippocampus and the perihippocampal region, and across perirhinal and perihippocampal region. So in a normal rat behaving normally in a task, we see coherence in this theta frequency band. We also know that this coherence goes up when the animals are looking at objects. So we think we are very hopeful that we'll see this begin to break down in our hydrocephalus rats. And this is just theta in a rat. You can kind of see it coming on. So this is a theta frequency band. You can see it right there. And now it's gone. And I think we have one more good bout here. At some point. There we go. So now we see theta again coming here. You can see it, and there it's obvious in the spectrogram. So we have a long way to go, but things are going very well. Oh, I also want to show you that, of course, we want to see alterations of gait. I don't have any data to show you because we're trying to automate the analysis of this, but we do already see alterations of gait at 4 weeks. And so I'm going to show you, so what you're looking at here is a rat walking across from underneath. You'll only be able to see the feet. And you probably won't be able to tell which is a hydrocephalus rat and which is the normal rat, because they don't actually, it's kind of subtle. But we did do a little bit of analysis, and what we noticed was that the stride length is a little bit shorter. This is a hydrocephalus animal, and the stance is a little bit wider. And you can look at them and see the obvious difference. Their feet are a little bit wider, their feet are turned out a bit. And so we've got gait, we have a memory model, and we think we're in good shape to try to make inroads into trying to ameliorate these memory deficits. And the idea we have is that if the coherence breaks down as the memory deficit develops, we may be able to restore theta coherence using optogenetic approaches. And then, so it's just one more time to watch the guys walk across the screen. And I think I already said all of that, so I'll stop there. Any questions?
You had color blocks, and this is just a technical question. Are rats in rats see color?
They cannot see color. So they have to be, those objects differ in luminance. Actually, when we project to the floor maze, we don't use any color because they actually see a little bit of yellow and a little bit of blue, but it's probably not behaviorally relevant. So I asked, can we get grayscale Legos? And my technician says no. We might have to make them ourselves.